Hey everyone, welcome back. I am back with another Bioinformatics Pro-Tip. We are staying today entirely inside our VS Code notebook, and we are going to learn a pure Python trick. Okay, so very common task in computational biology is comparing two DNA sequences to see if there are any mutations. If you have a wild type sequence and a mutant sequence, how do you find the differences? Okay, so today I'm going to show you the beginner way, and then I'm going to show you how a pro does it in a single line of code. So let's jump in. So let's define two short DNA strings. Basically, they are almost identical, but I have hidden a couple of single nucleotide polymorphisms in the second one. Okay, so what we will call them is sequence wild type and sequence mutant. Okay, I have prepared the sequences for both of them, so I'll use this for wild type and I will use this for mutant. Okay, if I ask a beginner to find the mutations, they almost always write a loop using range method. Okay, and it looks something like this. We can add a simple print statement to tell you, which is let's call it beginner method. Okay, and after that, what we will do is run a loop for I in range length of wild type. And what we will do inside this loop is if sequence dot wild type ith element is not equal to sequence mutant ith element, then we will basically I think we need to close this. Okay, and what we will print is uh, what we can say is mutation at index let's say I is equal to sequence wild types I uh, what we'll say is change to uh, sequence mutants I, right? And let's run this, and this works. Okay, it finds the T changing into a G, and the uh, right, and the T changing to a G later on. But in Python, using range is considered unpythonic. Basically, it's clunky, it's hard to read, and it's easy to make off by one errors. Okay, so the pro way is like professional Python coders use a built-in function called zip. Think of literal zipper on your jacket, right? It takes the left side, takes the right side, and physically locks them together pair by pair. So in Python, zip takes our two DNA strings and pairs up first letter with the first letter, the second with the second, and so on. We combine this with the enumerate function to keep track of the position index. Okay, so let's copy this, and what we will call it is the proway. Okay, so what we will do here is uh, for I, then we will call it base 1, base 2 in enumerate. We'll use the zip command here, sequence wild type and sequence mutant. Did we make any mistake anywhere? No, I don't think so. And if base 1 is not equal to base 2, we will print what we will print, mutation at index I base 1 base 2. And look how clean that is. Base 1 and base 2 are paired together directly. We don't have to deal with messy brackets like sequence wall type I and all this, right? It reads like plain English for the index and two bases in the zip sequences. If they don't match, print them. Okay, but we can go one step further. What if you don't just want to print them, but you want to save these mutations into a list for downstream analysis, right? So we can condense that entire loop into a single line using a list comprehension. Okay, so what we will do is we will write it in an ultimate one line. So we will call it mutations is equal to what we will write here is I B1 B2. Okay, uh, for I uh, B1 B2 in enumerate zip sequence wild type sequence mutant, and what we will add here is if B1 is not equal to B2, and let's call this what should we call it? We'll call it uh, one liner result. Okay, and what we will print is basically just mutations, and let's run it, and boom. In exactly one line of code, we zipped the sequence, filtered out the matches, and created a list of tuples containing the position and the two bases. This list is perfectly formatted to be dropped right into a pandas data frame, and that is your pro tip for the day. Next time you need to compare two strings side by side, ditch the range method and reach for the zip function. It will make your code faster, safer, and much more professional. Okay, so yeah, drop a like if you learned a new Python trick today and make sure you're subscribed, and I'll see you next time.